Hey explorers, need some more culture in your lives? Let's talk about bacteria, microscopic creatures that live on and in almost everything on this planet. Bacteria can be good, like the ones that live in our stomach and help us digest food. Or bacteria can be bad, like ones that make us really sick. Now, when I say bacteria are everywhere, I mean everywhere. But most are very helpful to us. Today, we're going to grow some bacteria cultures started from samples all taken from household items. We bought this great kit from Seaweed Solution Laboratories, which has everything we need for this experiment, including agar, cotton swabs to collect our samples, and Petri dishes to grow our cultures in. Agar is a jelly-like substance we get from red algae, a very common water plant. This becomes a food source for the bacteria as well as a stable place for it to grow. Growing samples in agar is a great way to see bacteria in action because even one from a little sample can become a colony of thousands. You'll find a link to this great kit in the video description. And while you're there, take a second to subscribe to Clayton's Exploration Station. You'll also find some links to follow our social media adventures as well so you don't miss any of the fun. The first step is to prepare our agar substrate. Following the instructions that come in the package, we mix the 8-gram package of agar with 350 milliliters of distilled water. We bring this to a boil either on the stove or in the microwave and give it a good stir so it's all mixed up. We set the agar solution aside to cool for 20 minutes. Then we put enough in each of our Petri dishes to cover the bottom with just a very thin little layer. We don't need any more than that, so you can discard whatever's left over. Put the lids back on your Petri dishes and set them aside for 20 minutes to continue to set. Have you ever heard of penicillin? This life-saving antibiotic was actually discovered by accident in 1928 by Sir Alexander Fleming. He was studying Staphylococcus bacteria, which is responsible for a wide range of infections. He had it in a Petri dish and forgot it when he went on vacation. When he returned, he found a mold growing around the bacteria which was stopping it from spreading. He soon identified that the mold made a chemical that could kill some harmful bacteria. He called it penicillin, and it's still very widely used today. With our Petri dishes ready, we just need one more thing: bacteria samples to grow into cultures. We do this using the cotton swabs that are included in the kit. Take one of the cotton swabs and run it along the surface where you'd like to collect bacteria from. Then, gently scrape the cotton swab through the agar in your Petri dish. Seal the Petri dish with a little tape, and if you're like me, label it so you know which is which. We've selected a number of household items so we can see the bacteria that grows all around us. I'm both very excited and a little worried to see what we're going to grow. We're testing a toilet bowl handle, the leaves of a plant, my cell phone, Walter's teeth, Daisy the cat's ear, a guitar string, and doing a comparison between a used water glass and the very same water glass right after it comes out of the dishwasher. I'm quite excited to see that comparison. The only thing left is to leave them in a nice warm area for 2 to 3 days, and then I get to employ one of my favorite scientific tools, observation. Observation focuses on watching what happens and making notes. This is very important because even the most well thought out experiments can go in unpredicted directions. In order to be able to repeat the process for research, scientists need to be able to see what happened and why, which all starts with observation. This could mean drawing a picture or taking photos of your samples, or noting down differences in colors, shapes, or anything else that you notice while your cultures grow. The more detailed the notes, the better.
Science!
Our bacteria cultures took a couple days before we started seeing any real growth, but after that, they didn't stop. By day 3, we were seeing yellowy green spots near where we had rubbed the samples on the agar, and by day 4, those spots had grown in size and had dark centers, which meant a higher concentration of bacteria. Some of the best results we got came from the bonsai tree, which is to be expected when you're taking results from living things. The sample we took from Daisy the cat's ear showed similar growth, which is a great thing because there are mites and bacteria that live all over living creatures, so it's actually nice to see. My guitar strings definitely need to be changed after seeing what grew from those, but the toilet bowl handle and cell phone were surprisingly clean. And Walter's teeth? They must have been spotless because almost nothing grew from that sample. I guess he really does focus on keeping his mouth clean and cavity free. I was very excited to see the comparison between the used water glass and the same glass after it came out of the dishwasher. The used glass did have some bacteria on it, but the very clean one looked like it did something to the agar. It started to curl up around the edges as if something in the cleaning detergent was killing the agar. I was surprised and quite happy with the results of my experiment. It was really nice to watch these cultures grow over a few days. I definitely recommend getting a kit of your own from Seaweed Solution Laboratories, and again, you can find that link in the video description. You do need to be patient, but after a couple days, you'll start to see some amazing bacterial growth. I'm even going to leave mine out for a couple more days just to see what continues to grow. When you're done with your experiment, wrap your Petri dishes without opening them in newspaper and throw them away in the garbage. If you're really keen to see observation in action, try it again using the same samples just to see if there's any differences between the first time and the second time you do it. Growing bacteria cultures is a great way to show us that the world isn't just really big, it's tiny and growing. And wash your hands, please, because it's very important. So clean up, keep detailed notes, and stay curious, explorers.